Hello, this is Mr. Philippek, and this little review video is on incomplete dominance, another form of dominance that we need to talk about in genetics. And now with incomplete dominance, the heterozygous is going to show a blend. Now remember, with simple dominance, the heterozygous shows the dominant phenotype, but here, since the dominance is incomplete, we're going to get something that's in between both the dominant and recessive allele. And so in this case here, I've drawn up a key where let's say that big R equals red, little r equals white. The heterozygous is going to show a blend between these two, and it's going to be pink. Now, how would you know if something was incompletely dominant? Well, typically in the problem, they're going to tell you. And so they'll say something like red is incompletely dominant to white, and the resulting offspring, the heterozygous is pink. That should clue you in. Other times they may say, well, when we cross a red and a white flower, here's what happens. We get red, white, and a pink. But anytime you see a blend, you'll know that it's incompletely dominant. So what happens here if we cross a red and a white flower? Well, here's our red, and remember now with incomplete dominance, the only way to get a red flower is that these alleles, or the genotype for this first organism, has to be homozygous. And then here's our white flower, and if we cross them, we notice we get 100% pink. All right, and so here we took a red flower, we took a white flower, and we got 100% pink. We don't even have red or white. Well, what happens if we take it a step further and we want to cross two pink plants? And again, here's our key. Big R is red, little r equals white. Homozygous is pink. So let's cross two pink. And now what I've done for you here is I set up the Punnett Cross, is just recolored them to show you that the big R is going to represent red, little r equals white. And when we cross them, we notice here that we start seeing what we saw way back in that first or that parental generation, where we see the reemergence of both red and white plants. And so at this time, let's go ahead and figure out the phenotypic and genotypic ratios. And so it looks like the genotypes we have are big R, we have big R, and we have little r. And so now let's just count up how many of each we have. We have one big R. We have two pink snapdragons, and then one little r. And I said snapdragons because that's typically the type of flower that we study in genetics that shows this incomplete dominance. And so what's interesting as well is that this genotypic ratio is going to be the same as the phenotypic ratio. Because right underneath here, let's write down what each of these colors are. So here we'd have one red flower, we'd have two pink, and then we'd have one white. And so with incomplete dominance, the phenotypic and genotypic ratios are typically the same. And so it's very easy now to figure out probabilities. So if we cross two pink plants, what is the probability of getting a red flower? Well, we just take a look here, and we see that that probability is 1 out of 4. What's the probability if we cross two pink plants that we end up with pink plants? That's going to be 1/2. And how did I get that? Well, we have 2 out of 4, and I just reduced that down to 1/2. And finally, what is the probability of getting a white flower? Again, that's going to equal the same as the red here. We just take a look. We have 1 chance out of 4. So if you notice here with incomplete dominance, there is no way to get true breeding pink plants. And so let's say if you ever got a call from somebody and they said, hey, I wanted nothing but pink snapdragons, and I don't want you to grow the plants, I want you just to send me the seeds. And how would I do that? I would cross a red with a white, and as you saw in our earlier example, we got 100% pink. Well, I hope this was a nice little review on incomplete dominance, and I hope it helps you out. And as always, thanks for listening.